Much of our semester is going to be spent talking about bacteria and to some extent viruses, and rightfully so, as they are kind of the exciting parts of microbiology. Module 3, however, gives us a little survey of the other microbes that are out there, some of the eukaryotic microbes and some of the things that they do as well, and some of them produce some very significant disease. Okay, so we'll start a little bit by talking about bacteria, just a little bit, and then we'll focus on some of the other microbes. All right, let's talk a little bit about prokaryotes and some of the things that they do, right? Because they're so vitally important to us. We say this all the time. Prokaryotes are everywhere. Yeah. They can do almost anything. Literally, they can eat so many different food things, things that are really weird to us. They can eat crude oil. They can eat pretty much any carbohydrate that's out there. They can live off radiation and things like that. They can live in the weirdest places. And really, there is no life without prokaryotes. We always talk about how plants are so vitally important. And of course, plants are important, right? But prokaryotes can produce more oxygen than plants. They produce all usable forms of nitrogen. Like literally, all forms of nitrogen are dependent on microbes, on prokaryotes. They're important for nutrient cycles. So whenever things die, whenever things need to get recycled, it's prokaryotes that play a vital role for that, right? And even for us, they help us digest our food. They provide nutrition for us, all this kind of stuff. Metabolically diverse, right? So we can do so many interesting things with them, such as clean up the environment. And we call this, they're genetically moldable, right? So we can employ them to do so many interesting things for us, including make our medications. Talk just a little bit about the nitrogen fixers. I did say that they produce all types of usable forms of nitrogen, and that's due to these guys right here, these nitrogen. How come this is not working? Oops, I gotta turn it on. Due to the nitrogen fixing bacteria, right? And maybe some of the most famous are the rhizobium. Notice, by the way, it's capitalized, right, and italicized. That means this is a genus, right? But there are a group of bacteria we call the rhizobia, right? Of which rhizobium is a genus, right? So in that case, rhizobia would not be italicized. Anyway, so they live in the root nodules of legumes such as clover, right? And what they do is they have this relationship, and we'll talk about how we can define this relationship, where they help convert this atmospheric nitrogen, which is like 80% of our air, right? But not usable for life, right? But they can take that and convert that to ammonia. And then the ammonia now is what we call organic nitrogen. And this function, this fixing of nitrogen, we'll talk about what fixing means later this semester, but this fixing of nitrogen is a vital function for all life, right? So we are 100% dependent on this group, the nitrogen fixing bacteria, to produce our ammonium for us. Life requires this. Of course, prokaryotes, and we're going to talk about this a lot, the pathogen, right? The disease-causing organism. Remember, less than 1% are human pathogens, right? But because they have these huge human impacts, they're much of the focus of our semester and much of the focus that the general public has towards the microbes. They don't care about the ones that are recycling elements for us. They don't care about the things that produce therapeutics and things like that. Really what they care about are the ones that cause disease, right? And rightfully so. Of course, getting sick sucks, right? We've all been sick before, so that's not a good thing, right? But then, the impacts on the financial impacts that we have, just the general impacts on our infrastructure, it has this huge effect, right? And so that's, of course, why much of our study of microbes really is geared towards preventing, treating disease and things like that, right? And then they do other things like food spoilage, right? And believe it or not, they do cause disease and other things that are not humans, things like plants and animals, and all of these things combine to have like a major impact on humans. So when we talk about microbes and the relationship with its host, right, we can define them based on these types of interactions, right, depending on really who benefits, who is harmed, the nature of this relationship, right? So the term symbiosis simply means interaction between species in a community. It can be one and one, it can be group and group, right? So really that's all this is. In many ways, symbiosis simply means living together, right? And we'll often use, these two have a symbiotic relationship, right? But the term symbiotic relationship is actually kind of vague, right? Because you and your roommate, you guys have a symbiotic relationship, right? Because you guys live together, you and your family members, and things like that, right? So what you want to do is you want to define who or what happens to each of the populations in this relationship, right? And of course, the best type of symbiotic relationship is something called mutualism. And in mutualism, we'll just say population A and B, right? Both populations benefit. So they both get gain from this, right? So let's go back to this whole nitrogen fixing bacteria. I said they're found in the roots of legumes, right? Things like peas, beans, clover, things like that, right? That's what we call a mutualistic relationship, because the bacterium, remember, produces this nitrogen for the plant, right? And because it's intimately growing in the roots of the plant, like literally in the roots of the plant gains this immediate nitrogen, which is such a limiting factor in soil, right? But at the same time, the bacterium is protected, right? The plants kind of, the roots kind of wrap around the bacterium, right? And then it gets like all this runoff of carbohydrates being produced by the plant, and then this protection from predators and things like that, right? So it's this really beneficial relationship for both organisms, right? So this is what we call mutualism. In amensalism, one population is harmed, the other is unaffected, right? It's kind of like a pathogenic relationship in which the organism causes like an effect on its host, right? So the host is harmed, right? But it's a little different in that the pathogen in this case would not really gain anything from it. It just kind of happens to cause disease. A good example of this is like a plant relationship, right? Whereas you have this one plant that's growing, and then its leaf causes a shade to cover another plant, right? And then that plant would be affected or be really harmed by that because it doesn't have access to the sun, right? The first plant isn't gaining anything from this, right? It's just doing what it does, right? But it's harming a second plant. A microbial example would be this microbial production of antibiotics, right? And there are many molds that can produce antibiotics. The antibiotics end up killing or harming the bacterial population around that, but for the most part, the fungus doesn't gain anything directly from that, right? It's this sort of indirect benefit in that it's kind of killing off its competition, right? But it really, truly doesn't gain from this relationship. In commensalism, you have this relationship where one microbe benefits, the other one is unaffected, right? And many of our microbes that live on us are what we call commensal. They hang out there, they kind of do their thing. They gain the protection from us, especially if they're inside us. But we don't necessarily benefit from them being there. They're just kind of there, right? But they in turn, because they're inside, it's always warm. They got these really good, nice environment and access to some of the food sources that are present inside, right? So, they just kind of hang out there. We like them being there, right? They're kind of important to us, right? But in general, there's nothing specific that they do that helps us out. In many ways, we could consider some of these as well to be neutralism, where really there's really no benefit either way, right? The ones maybe on our skin, they're just there, right? They're not really doing anything for us, right? They're not really harming us. We don't really notice that they're there, so there's this meh type of relationship, right? Where, we don't really care, right? And so that's this whole idea of neutralism. And then of course in parasitism, the worst kind is where one organism benefits, in this case the pathogen would benefit, whereas the other organism is harmed, okay? So again, symbiotic relationships, a lot more complex than we make it out to be, right? Living together, right? But really there's going to be an effect on each type of population. Collectively, when we talk about microbes associated with within or around a larger organism, we call that a microbiome, right? This whole ecology of microbes that surround this larger organism, right? And we have this intimate relationship with our microbiome. And then we can have these resident microbes that are living there permanently, and then transient, which are kind of temporarily there, right? And then things like diet, age, and all this kind of stuff can change the constituency over time, including the resident ones. We're going to be born with certain microbes, and especially within our gut and within our skin, and over time that can change, right? But we kind of define the resident ones as being there for a relatively long time, months, years, that kind of stuff, whereas transients are just sort of temporary passerbys, just kind of on the order of weeks. Okay, so that's enough for bacteria for now. We're going to get back and talk about bacteria plenty, right? Let's talk about some of the other types of microbes, and we're going to spend a little bit of time talking about our eukaryotic microbes, right? And we're going to start off with the protists. Just quick review. Remember, protists are eukaryotic microbes. As a group, they're extremely diverse. So many different morphologies, so many different life cycles, nutritional needs. They're really hard to define. Oftentimes they're kind of like this miscellaneous group of microbes that really didn't classify well anywhere. Most are free living and benign. They just kind of do their thing. They're part of the food cycle. They eat bacteria for a living, and then they themselves get eaten by larger organisms, right? So they're very important in the ecology. But of course, some are human pathogens, right? And probably the most important protozoan human pathogen is Plasmodium, which causes malaria. And malaria is this important worldwide disease. We don't think about it too much because of the fact that, we don't have incidents of malaria around here. But malaria is quite important worldwide. Giardia as well causes some intestinal stuff, right? So, there are many protozoan parasites that we will talk about. As a group, protists are not a true phylogenetic group. Remember, phylogeny means that groups that share a common evolutionary origin, right? So the protists, they don't really share an evolutionary origin. In fact, we have these animal-like protists, these plant-like protists, the fungus-like protists, right? But because they're all single-celled, mostly, they're all kind of doing what they do, and they don't really fit in the animal or in the plant, right? They said, you know what, let's just have this one large group called the protists, and we'll chuck them in there, right? So often within protists, we'll refer to these as these kind of sub categorizations, right? And these would be more phylogeny. So our protozoa would be our animal-like protists, right? They're unicellular, they're motile, like little animals. Algae would be our plant-like protists. Our slime molds and water molds are fungus-like, right? And just like their larger cousins, more complex cousins, they do very similar things, right? So our animal-like ones are protozoa. They move, they eat, they poop, they sense and respond to environments just like animals do. The algae, they're photosynthetic. Some of them are unicellular, some of them are multicellular, right? But they are photosynthetic, which of course is one of the key features of the plants. Our fungi, slime molds, and water molds, they're decomposers, right? So again, very important part of the food chain. And then mostly unicellular, right? But there are some multicellular slime molds and water molds. Often we refer to these as plankton, right? So the protozoa can often be called zooplankton. Algae are phytoplankton, and typically we use this term when they're in some sort of aqueous, usually marine environments, right? So often you'll hear these things: zooplankton, phytoplankton. It just means the same thing. Protists, when we're talking about especially the pathogenic ones, have very complex life cycles, right? And because they have this interplay between hosts, and then each protozoan parasite, each protista has its own very complex life cycle, right? But we're going to talk just very specifically about some of the pathogens. Now, within this life cycle, it undergoes many changes in form and shape, right? So there's a couple of things I want us to kind of know. The trophozoite is our feeding and growing stage of the life cycle, right? So whenever you talk about the trophozoite, you're talking about the form that's like actively growing, actively dividing. The cyst is a cell with a protective coat, right? In many ways, you can think of this kind of like an endospore, maybe more like a seed, right? So, it's really depends on how you want to define this thing, what's going on, right? So let's call this like kind of like a seed, which is, for reproduction, kind of like an endospore, not really reproduction, but, sort of this whole protective coat, right? That then allows it to survive passage. And we'll talk about some protozoan parasites where the cyst stage is quite important. Occasionally, you'll see this referred to as an oocyst, right, which is kind of like an egg cyst, right, a cyst around the egg form, right? Then, for us, we're going to keep it simple. We're just going to say an oocyst and a cyst are more or less the same thing. You'll hear the terms encystment, right, which is the formation of the cyst, and the excystment, which is a release from the cyst, right? And many protozoans or Many of these parasites are capable of both sexual and asexual reproduction, which is very important in the life cycle, right? Because asexual reproduction is fast and easy, like really allows the parasite to get its numbers up really quickly, right? Whereas sexual reproduction is important for things like genetic diversity. You need to have a diverse gene pool to really survive. Okay, so rather than talk about like general things, we'll kind of use some of the just some examples of protozoan parasites as a way to kind of highlight some of the things that protozoans do, right? So here we're going to talk about EIMERIA, which is a genus of an organism, a protozoan, that causes a disease called COCCIDIOSIS. Now, EIMERIA can cause disease in animals, cattle, dogs, cats, humans, things like that. And, this one here is highlighted because of its prominent cyst stage, right? So it has a cyst stage that's it's necessary for it to cause this disease, right? But unlike some of the other organisms that we're going to see later, it's a one single host, right? And it's usually a specific host. So the specific species of EIMERIA will really only be able to infect its preferred host. Let's see. So it begins with when the cyst here, in this case it's an oocyst, is swallowed by the gut, right? And enters via some sort of ingestion. And it's this oocyst, right? Remember the cyst that's protected, right? And here is this, what we call the environment area, right? So here it's got to spend time outside. It requires a lot of time for this stuff to happen, right? It's cold outside, not a lot of food, right? So the cyst allows it to survive this passage while it's just kind of hanging out, waiting for the animal to come by, right? And then when the animal ultimately ingests it, it causes this disease, right? It's specifically things like it's intestinal, so things like diarrhea, dehydration, when it gets really bad, weight loss and death, right? And in the meantime, it's undergoing, and notice the trophozoite here, it's growing actively, right? And doing its asexual reproduction. It's really just growing real fast, increasing its numbers. And then at some point, as it's kind of reached its peak, it then goes and develops this cyst form, right? So it undergoes encystment as it does its sexual reproduction, right? And then once it's formed its cyst, then it gets shed out in the feces, right? And then once again, it's a cyst that's so important. Now that it's done with this host, it needs to spread to the next, right? So the cyst stage is very important in EIMERIA being able to pass from one to the next host, right? NAEGLERIA, it causes a disease called primary amoebic encephalitis, right? And the word encephalitis simply means it's a swelling of the brain, right? It's often known as brain-eating amoeba. And I like this one here because of this really interesting thing that it does, right? It does this amoeboid movement. Actually, it just kind of lives there. NAEGLERIA is what we would consider to be quite a poor pathogen. It doesn't really do things very well. It doesn't really, you could be surrounded by it. It really doesn't affect you too much, right? But if for some reason it gets in through your nose, and usually it's not even just in your nose, usually it's like way up in your nose, right? It then uses its amoeboid movement, these pseudopods or pseudofeet, which just kind of walks and crawls, right? And makes its way to your brain and then starts to destroy neurons, right? And it's extremely deadly if you ever catch this, right? This brain-eating amoeba, because there's no real treatment for it, right? It's hard to treat. It's up in the brain, right? It's you can't just like open up your brain and cut it out, right? So it's quite a deadly disease, right? But typically you only get it in when you're playing in warm, fresh waters, right? And the most common way to get this, it's like water skiing. So you're out there water skiing, and then like you fall and you eat it, right? And then it happens to like just jam this thing way up in your nasal cavity, kind of puts it near its brain, and then it lets it do a thing. Extremely rare disease, right? But again, very deadly if ever contracted. Trypanosoma causes this thing called sleeping sickness. Very common in Africa, right? African sleeping sickness, right? And the African version uses the tsetse fly as its vector, we call it, right? The vector is what spreads this disease from one host to the next, right? Now, the thing about the vector is it's actually a host, right? So Trypanosoma is a really good example of an organism that uses two hosts, right? Both the humans and the tsetse fly. right? And it's this tsetse fly that through the bites then transmits the trypanosoma pathogen, right? Let's see. Initially, it takes a blood meal, right? It bites the human, and then it then multiplies, does its asexual reproduction in the host, attacks the lymph nodes, right? Typically, you get these swollen lymph nodes. That's kind of characteristic of sleeping sickness. And then this fever, pains, and then this mass itching, usually around the site of the bite, right? But as it starts to infect the nervous system, you get this really weird disruption of your sleep cycle, right? And so that's where the name sleeping sickness comes from. Just this really weird disruption of your sleep cycle. Sometimes you can't sleep. Sometimes you just sleep for way too long. And you get this really weird confusion. And then again, if it gets really bad, weakness and death, right? Now, the trypanosoma doesn't want to stay in the human. It needs to complete its life cycle, right? So in order to do that, it waits for another tsetse fly to come and then bite this infected human, and then it can migrate to the tsetse fly. And in the tsetse fly, then it undergoes its sexual reproduction, right? And does its thing, and again, waiting around in the salivary glands until the next human gets bitten, right? And it's this interplay between the two hosts where it has to go. Humans cannot pass on trypanosoma to other humans. Tsetse flies cannot pass it on to other tsetse flies, right? So this interplay, and this is this whole really complex life cycle that we were talking about. So those three kind of highlight some of the things that some of the protozoan parasites can do. There are a lot of interesting protozoan diseases. There's just not enough time to talk about them all, right? Just to go through a list of all the diseases. So here's kind of a list, for those of you guys, are interested in some of these diseases for your disease profile project, right? You can, here's some very common, interesting diseases. Toxoplasmosis, it's kind of a neat disease. It's carried by cats, right? It really kind of messes you up, right? It's really neat stuff. Malaria, extremely important worldwide disease. Chagas disease is American sleeping sickness, right? It's caused by another species of trypanosoma. The cool thing about this is, it's kind of disgusting, is that so it's not a tsetse fly, it's a different insect, right? It's called a kissing bug, and it gets its name kissing bug because it likes to feed around the mouths of the humans while you're sleeping, right? So it'll sit there and it's kind of like it's kind of kissing you while you're sleeping, right? I know, really gross, right? Some of these other diseases, trichomoniasis is one of the very common STD. It's estimated that almost 4 million people carry this thing worldwide, right? But only 30% show symptoms, right? So it's this very important STD. Typically we think of them as being bacterial or viral, but in this case it's caused by a protozoan. Amoebiasis is an incredibly important protozoan intestinal illness, right? They say about 480 million people are infected. Got about 40 million new cases worldwide, right? And of course, a lot of these are due to are found in areas where the sanitation is quite poor, right? So in many developing countries. All right, so moving on then from protozoan, well, let's talk about parasitic helminths, right? And if you thought the protozoans are gross, this is the kind of stuff that really gets you the oogies, because we're talking about the worms, right? Nematodes are roundworms, platyhelminths or flatworms. There are many types of helminths that are out there doing their thing, just like protozoans, just living, right? But of course, we care about the parasitic ones, right? Because they're the ones that are going to, again, have major human impact. Parasitic helminths have a lot of characteristics that differ from free-living helminths. Generally, they lack a digestive system, right? So they don't have a really complex digestive system. They're kind of dependent on the host doing the digestion for them. They have a reduced nervous system, right? Because they don't have to be out in the environment very much. They just kind of live, again, within this very confines of the human host, right? So they're not all about sensing and responding to environments, right? They don't often move too much, right? But they have a very complex reproductive system. So just like some of these protozoans, very complex life cycle, often interplay where they have succession of hosts. One host leads to the next host. Sometimes they'll use each host for different types of reproduction. Now, the type of reproduction that these helminths can do depends on the type of organism, right? So we have this term dioecious, which means that they have male reproductive organs in one, female in another, right? So some species are dioecious, right? In this case, these cases, we're talking about sexual reproduction only, right? Whereas monoecious or hermaphroditic worms, they have both organs, right? So in this case, they can actually do asexual reproduction in the event that a suitable partner does not exist. All right, let's talk a little bit about each type, right? So the nematodes are the roundworms, right? So they're unsegmented roundworms. Generally, they do have a complete digestive system in the fact that they have a mouth, intestines, and anus, right? But again, they kind of really helps, or they like being in the host because the host does a lot of the digestion for them. Enterobius, aka pinworm, is the most common infection in the US, right? And it's kind of, again, you know, like I said, these things kind of give you the oogies, right? So I hope it doesn't get too weird for you, right? But a pinworm causes this itching around the anus, right, as it's starting to leave. You know, very similar way if you guys ever seen dogs kind of scooting, right? And they're scooting because they have like worms coming out of their exit hole, right? And it causes a little itching, you know. Same with pinworm. As the worms are itching, they're just kind of crawling around the exit hole, right, and causes this itching, right? And it causes a really, again, I don't think I need to talk more about this, right? Trichinella. I think many of us have heard of trichinella, right? It comes from eating undercooked pork, right? And for the most part, one of the reasons that pork is generally frozen prior to selling in the store is because it's the freezing process can kill trichinella, right? So typically nowadays, you really only get trichinella from pork that you catch, i.e. the boars that you hunt. The platyhelminths are the flatworms, and the flatworms come in two forms: the flukes and the tapeworms. Flukes are like these flat leaf-shaped things. They have like this oral sucker right there, right? What they use for attachment, right? And typically flukes are named after the tissue that it infects. So a lung fluke infects a lung, heart flukes infects the heart. Tapeworms are segmented flatworms. So they're a little bit different from the flukes and the nematodes in that they're segmented. So they have these little kind of regions, right? And each region can have kind of exist independently. So if you cut them, typically they can regrow. And then just like flukes, they use these suckers and sometimes these hooks for attachment, right? So they often hook, stick into your intestines, and they kind of do their thing, right? So just like with our protozoans, we're going to talk about a couple of these, the helminths, right? And just talk about the life cycle. Schistosoma is a very important disease worldwide, right? Sometimes called snail fever because of the fact that it lives technically, we get it from snails, right? So it's a parasitic flatworm. The eggs are released from the snails, right? So where's the snail? Blah, 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 blah. Eggs hatch, right? And then so eggs are released from the snail, and then they are free swimming in the water, right? And then you get schistosomiasis when you ingest it, right? So it's typically playing in the water that it's in, and of course in areas where you don't have clean water, you're dependent on the river waters and on this kind of stuff to do things like your laundry, to cook in and things like that, right? And if they're not properly treated, then you're going to end up ingesting these things, right? And then it causes things like abdominal pain, diarrhea, bloody stool, right? And guess what happens? Guess how these things leave? Yeah, it's through the feces of the human, sometimes the urine, right? And so it's this expulsion. And again, as with the areas that they don't have proper sanitation, right? The eggs make their way back into the water, and then they then are able to attack the snail and then do their things in the snail, right? So again, this whole interplay where one, they do this asexual reproduction, and then the other, they do the sexual reproduction, right? So this important interplay, and they have to complete this life cycle. It doesn't go from human to human, doesn't go from snail to snail. Taenia is this disease called TAENIASIS, right? It's a pork or beef tapeworm. And it's contracted by eating the live worms, right? So they're really living in just the pigs or the cows, right? Hanging out in the muscles, kind of doing its thing, right? And then humans can get it when we eat raw or undercooked meat that's been infected, right? So these are things that, of course, in the industry are going to be screened for quite often, right? So typically nowadays, we only really get them from facilities that are quite negligent in keeping up with their screening, right? Or from wild pig, wild cattle. Symptoms are generally mild, right? Little bit of weight loss, some abdominal pain, and typically the pork tapeworm tends to be often more severe. I included this one here because it has like this this urban legend, right? So back in the day, there's a lot of historical references to people on the tapeworm diet, right? And so as the story goes, people would eat these cookies to help them lose weight. And what the cookies contained were the eggs of the tapeworm. Typically, it's never really been identified, but it's believed that it's TAENIA, where then it grows and multiplies. You can eat what you want, right? And then it's out there doing its thing in your abdomen. Maybe get a little bit of pain, right? But for the most part, you get this weight loss that kind of comes with it. Of course, because it's taken all your nutrition, right? And then the idea is that after a month or so, you're supposed to take like a second cookie, right? And that cookie has now the medication to get rid of the tapeworm, right? But some people, they were loving this weight loss so much that they didn't take that second cookie, right? But then, of course, if you don't get rid of it, then you really start to suffer from things like malnutrition and stuff like that, right? Believe it or not, people still do this nowadays, right? Apparently there's some treatment. I think you can go to Mexico and spend thousands of dollars for tapeworm diet treatments, right? I think I saw an article somewhere where someone in Iowa tried it, right? Because I guess they were just really wanting to lose some weight, right? So, the FDA has to come out and say stuff like the tapeworm diet is illegal. You have to be told this kind of stuff. Isn't that crazy? But I don't know. I guess for some people, vanity is quite an important thing, right? So I don't know about you. If someone says, hey, you can lose some weight by eating tapeworm, I'm like, I think I'll pass, right? But, for some people, it's like, it's a quick and easy way to do this kind of stuff. Of course, extremely dangerous. There are other common helminthic diseases, right? Ascariasis is probably the most common soil transmitted helminthic disease, right? I think they say an estimate of 1 billion people carry this thing, right? But the really cool thing about ascariasis or Ascaria, the helminth that causes the disease, right? And so whether it's a nematode, that's what this N refers to, is that these are probably the largest helminths, and these things can grow like over 1 foot in length. Isn't that crazy? Like this worm that's like over 1 foot long hangs out inside you. Madness. Angiostrongyliosis is better known around here as let's just write that in there, as rat lungworm, right? So we've had a lot of incidents of this on the big we're supposed to call it on Hawaii Island, right? So Hawaii Island has this endemic case of presence of Angiostrongylus, the organism that causes rat lungworm, right? So really interesting stuff. And the craziest thing that some people say about this disease is that they wouldn't wish this on their worst enemy. Some say that's this extreme impact, right? Extremely painful. And then oftentimes you can have lifelong effects of this kind of stuff, right? Okay, that's enough for the helminths, right? So we're now past the Uji phase, right? Now we're going to talk about the fungi, talk a little bit more about maybe the structures of the fungi. And remember, let's go back to this term mycology, right? The study of fungi, right? It's this whole MYC prefix that denotes a fungus, right? Fungi can be multicellular, can be unicellular or single-celled, right? And we've seen these things, right? We see the molds growing on your wall. We see the mold growing on your food. Those are clearly multicellular, right? But there are single-celled ones out there as well, things like the yeasts and stuff like that. Fungi are very important in the food chain. They decompose dead matter. They help recycle vital elements, right? And of course, we use them things for food, right? Mushrooms, for example, are fungus, right? So we eat these things. Cheeses and stuff like that are often produced by fungi. We eat those things, right? But they also help us make antibiotics, right? Fermentation, all this really good stuff. Ultimately, they do cause disease, right? And the MYCOSES, that will be plural, right? MYC, that's a disease caused by a fungus, right? Singular here will be mycosis, right? Quick review. Remember, fungi have cell walls made of chitin, right? So the chitin cell walls fungi have chitin cell walls, right? Kind of characteristic of fungi. And then they have something called ergosterols in the membrane. Remember, eukaryotic cells have sterols, right? Fungi have ergosterols, which is, more or less the same thing, but specific enough that if you can identify it, then you can classify it as a fungus, right? Fungi are capable of reproducing both sexually and asexually, right? Sexual reproduction is often using spores, right? But you can also they can also reproduce asexually, right? So again, the idea with asexual reproduction is that when they're growing rapidly, you want to do asexual reproduction, right? Because you can just really quickly, and they'll do this through mitosis. And then when you need to develop some genetic diversity, then you can do some sexual reproduction, right? Where we get together, find a suitable mate. In this case, they call it a plus mating type, a minus mating type. Mix some gametes, right? And then produce offspring that are genetically distinct. Fungi can be classified or characterized, I guess, into 3 different types of fungi. The yeasts are unicellular or single-celled. They reproduce by budding, right? So it's mitosis, produces like these little, kind of imagine as like bubbles coming out of the yeast. Molds are multicellular, and we've seen molds, right? So the molds are multicellular masses, and we'll talk about the molds very soon. And the dimorphic fungi can actually do both, right? It can grow as both a yeast or a mold, and really depends on the environment. And many pathogens, many fungal pathogens can be dimorphic, right? Where they can grow as yeast when the times are right? Typically as when they're pathogenic, they'll grow as a yeast because they can do that really fast, right? But then sometimes when things are not so good, they'll grow as a mold because the multicellular growth allows better survival of the organism as a whole. So speaking of molds, multicellular growth is done by growing filaments, right? So we have these long filaments that grow, and each filament is called a hypha, right? Hyphae for plural, right? So each hypha then just grows and elongates, right? And it's very important for the fungus to be able to do this because, this extending of filaments allows it access to food sources that are farther out, farther away, right? So then once again, allows for survival of the organism much better. Hypha can be separated by these walls. This is what we would call a septate hypha. Or we can have non-septate hypha, also known as cenocytic, right? So again, it's very characteristic of the fungi, whether they form these divisions into these cell-like units, these septate hyphae, or the non-septate hyphae, the cenocytic, right? Occasionally what you're going to get is something called a pseudohyphae, right, where cells just kind of clump together, right? They're not really hyphae, right? They have to kind of elongate, right? But, because they're sort of multicellular, they've been given this name pseudohyphae, right? Now, as we have a bunch of hyphae kind of growing together, that's often called a mycelium, and that's because the filaments are just so long and so massive and so intertwining that it's really hard to distinguish each different fungus, right, or the thallus. Now, thallus would be the body of a fungus. Each individual organism would have a thallus, right? But they all kind of grow amongst each other, right? So this whole network is called this mycelium. Fungi are grouped according to things like their life cycle, their sexual spores. There are 7 major groups, right? But depending on which classification system you use, sometimes they'll have a little bit more, right? But we're only going to talk about these 3, the Zygomycota, ASCOMYCOTA, BASIDIOMYCOTA. And again, notice MYC, right? Referencing the fungus. And we'll just talk a little bit about some of the major characteristics of each of these groups. The Zygomycota produce cenocytic hyphae. They use something called zygospores for sexual reproduction, and sporangiospores for asexual reproduction. So these guys are actually characterized by these long sporangia that kind of extend up, right, off the surface, and then have their spores in this sac, right? And that's what the sporangiospore is, right? So these things coming up, things like rhizopus, which is what we call a bread mold, right? So typically the white fluffy thing growing on your bread is rhizopus, right? So it kind of grows up, and then it has these little black sporangia or at the top, right? Each sporangium contains hundreds of spores, which then will pop and then burst. Okay. Mucor is a very important Zygomycota. It causes necrotizing infection, right? So it's just this very rapid, causes tissue death, right? So in the systemic infection. The ASCOMYCOTA are in this case characterized by a septate hyphae, right? So they have these divisions, right? And they use ascospores for sexual reproduction, conidia for asexual reproduction. And so they're characterized by this whole naked cluster of spores, right? So the sporangiospores, the zygomycota have this sac around their spores, right? Here they have just kind of everything in chains. Aspergillus is such an interesting genus, right? Because Aspergillus can cause allergies. So many people are allergic to Aspergillus spores, right? They cause a lot of neural infections, right? Aspergillus flavus produces this really powerful neurotoxin called an aflatoxin that contaminates nuts and grains, right? But some species of Aspergillus provides food for us. So shoyu, if you guys don't know where shoyu comes from, it's fermented soybean. And the thing that they use to ferment it is a species of Aspergillus, right? So I always find that fascinating. Like within a genus, right? All these related organisms, we have these guys that are extremely deadly, producing this powerful neurotoxin, yet its cousin we used to do and make maybe the most wonderful food product next to beer, shoyu, right? And can you imagine life without shoyu? I think we would riot, wouldn't we? Candida albicans is another very important ASCOMYCOTA. It's actually it's a commensal organism. It just kind of lives within us, hangs out with us, right? It's found in the vagina, it's found in the throat, in the intestines, right? Just hangs out there. But every now and then it can cause the disease, right? So we call this an opportunistic pathogen, right? So every now and then, when given the opportunity, it'll cause disease. Right, so it can cause yeast infections, vaginal yeast infections. Thrush is this infection of the throat, right? So it can cause disease, but for the most part, it doesn't, right? So it's one of these. Hey, sometimes we like you, sometimes we don't. Finally, the Basidiomycota have these club-shaped structures that produce spores within this thing called a basidiocarp, aka a mushroom, right? So the Basidiomycota are maybe the most famous in that, these are the things that we think of as a fungus. Oh yeah, fungus, mushrooms, right? I love eating these things, right? Very important decomposers, right? And whenever you see fungus, you know that there's a bunch of organics around because these guys are just going to be eating them up like crazy. Of course, not all fungi are edible, right? So be careful. Don't just look at a fungus and say, or a mushroom and say, we're going to eat this thing, right? They're not all edible. And then we have some very important basidiomycetes or Basidiomycota. Cryptococcus neoformans causes very serious respiratory infection, right? In fact, it's this disease where you often get by inhaling the yeast, right? This Cryptococcus. And it's often one of the most important or infection of AIDS patients, right? AIDS patients have a very reduced immune system, right? Typically it doesn't cause too many issues, right? But for AIDS patients, it's one of the major leading causes of death for AIDS patients. Amanita phalloides, aka the death cap, is an extremely powerful toxin, right? This poisonous mushroom, right? But interestingly, the toxin itself, because they know the mechanism of action, has been used to elucidate a lot of things in molecular biology, right? Because of the proteins that it attacks. So just like before, we'll talk about some of these. Ringworm, right? That's kind of a very common, especially here in Hawaii. It's quite endemic, right? Ringworm is not a worm, right? It's actually a fungal infection, right? But it's called ringworm because it forms this ring on your skin, right? And looks kind of like this worm that's been burrowing through your skin, right? But it's not a worm, it's a fungus, right? Athlete's foot, right? Sporotrichosis is kind of interesting. It's this also known as a rose gardener's disease or rose thorn disease, right? It's typically a gardener's infection. And so it's through pricks and through cuts and scrapes, which the fungus then gets into and under the skin, right? And in there, it causes generally some mild skin symptoms, right? But it can cause some more serious cases. In the case that you inhale it, it can get to the lungs. If it gets deep into your tissue, it can cause problem in your joints. But interestingly enough, feral cats are kind of carriers of this disease, Sporothrix. right? And it's actually considered an occupational hazard for veterinarians, right? As these cats, with their scratches and things like that, they can introduce the fungal spores into the skin, and many veterinarians can get sporotrichosis, right? Because of the fact that cats introduce it to them. Okay, so that's enough about fungi. Let's talk a little bit about algae. Right, algae are what we call autotrophs. They use inorganic carbon. All right, we'll learn more about that coming up next. Yeah, I believe it's next module. Yeah, good stuff, right? We're going to learn about photosynthesis, right? They're called phototrophs. They collectively, algae produce about 70% of oxygen and organics in aquatic environments, right? So they produce so much oxygen for us, right? And worldwide, there are many other types of algae that grow everywhere, right? That really contribute to this oxygen that we breathe. Just like many of the organisms we talked about, they can reproduce both sexually and asexually. They can grow single-celled. They can be enormous, as we will see. Typically, we classify them according to cell structures, according to the pigments that they use, right? There will be different types of photosynthetic pigments. So let's talk about some of the groups, some of the common groups of algae. Dinoflagellates are probably the most significant, maybe not. But they're definitely amongst the more pathogenic ones, right? Because of the fact that many of these are able to produce toxins, right? They're called dinoflagellates because of the fact that they have this cellulose armor, right? This kind of, look at this armored thing, right? Single celled, and they use this one flagellum to whirl. So it just kind of twists and whirls and moves around, right? So it's quite motile, able to do what it does. And some of them do something called bioluminescence or bioluminesce, which is this glowing caused because they have these organs that trap bacteria that are able to glow, right? So this is from your book, terribly grainy, by the way, right? But here's a shoreline, and way off in the distance, this glow is these dinoflagellates out there that are able to bioluminesce, right? Alexandrium is an example of one of these types of dinoflagellates that cause something called paralytic shellfish poisoning. Try to say that real fast, right? And then they produce this toxin, right? This really powerful toxin, which in and of itself isn't that big a deal, right? But every now and then, they have something called a red tide, right? They call it that because Alexandrium is red, by the way. It's that red pigment that it uses for photosynthesis, right? Every now and then, when the conditions are perfect for Alexandrium, and it just grows to this huge abundance, so much so that the ocean looks red, right? So we have a ton of these guys swimming around, and then as they settle, it then causes this buildup of the toxin, which then accumulates in shellfish, right? And this accumulation then, as you eat it, makes you extremely sick, right? So this whole idea of paralytic shellfish poisoning. And that's kind of a theme for a lot of these dinoflagellates. Seaweed, of course, are also algae. And, seaweed can be very small. It can be extremely large, right? And some of these large brown algae, these brown kelp forests, are hundreds of meters long, right? So here, I know it looks green, right? But that's because of the fact we're underwater, right? But we have these super enormous, long, tree-sized brown algae, right? We have these red algae and these green. And the reason that there are all these different colors is because they use different pigments for photosynthesis, right? And it allows them to live at different depth. The green ones are the ones that are closest to the surface. They're going to do photosynthesis very similar to plants. And then as we start to go down, I think red, 50-50, right? I think red are the ones that are going to grow deepest because the light kind of filters out. So some of the green light doesn't make its way down. So it has to use different wavelengths of light for photosynthesis. They're very plant-like, right? They have these leaf-like structures, these stalk-like structures, these special structures we call holdfast to attach to things, right? But they're not really like leaves. They're not really stalks. They're not really roots, right? And they are fully dependent on the water environment for them to go and for them to survive. That's pretty much all we want to say about algae, right? If you look at the theme of the diseases that algae cause, many of these are going to go and be very similar to the red tide, the Alexandrium we just talked about, right? A lot of this stuff is this accumulation of this toxin that then kind of builds its way up to the food cycle. I think maybe for us, the most significant of this is Ciguatera fish poisoning, right? Again, the idea is that Ciguatera is going to go and build up produce these toxins, right? They get eaten by these small little things, which then get eaten by bigger fish, right? And then as this big fish eats a bunch of these, it starts to accumulate, right? And then when the human eats it, well, then you get yourself some serious infection, right? Some fish poisoning. Lichen, I think lichen would be our last eukaryotic group, right? Of microbe. I don't know why I said right, of course. I know you don't, right? So the lichen are the last group we're going to talk about. Lichen are interesting because they're this mutualistic combination of an algae, right? An alga, singular, or a cyanobacterium, which is a bacterium, right? So you can actually have this, it's either an algae or a cyanobacterium and a fungus, right? And mutualistic. Remember what mutualistic means? Both organisms benefit, right? The alga produces food, right, through photosynthesis, so it'll photosynthesize, do its thing, produce carbohydrates. The fungus provides a holdfast or protection. It kind of wraps itself around the algae and then allows it to survive and just live in areas that really are unsuitable for either, right? So, and lichen then can, because of the fact that they're really not dependent on a food source, and because of the fact that the fungus allows it to stick everywhere, they grow in the weirdest places. Any surface, right? They can grow on the surface of plants, and anything that grows on the surface of a plant is called an epiphyte, right? So we're used to seeing them on things like rocks, on trees, on your fence, right? What is up with that, right? And they're slow growing because they're kind of dependent on this photosynthesis and really nothing else, right? There's not a lot of moisture typically where they live, right? But they can be slow growing because they're really not competing with anything else. It is interesting because they're technically two organisms, right? But really each one of these parts cannot survive without the other. However, they are more classified as a fungus, right? And the fungus that is part of the lichen is typically an ASCOMYCOTA or BASIDIOMYCOTA, right? And just like, we're going to use the same terms of a fungus, right? The whole fungus, the lichen itself is called a thallus, right? So that's the body of the lichen. The cortex is the kind of the fungal layer of top protection, but then we have the algae right on top, right? So the algae or the cyanobacterium lives right underneath that, right? Protected from the harsh environment, but close enough so then it can do its photosynthesis. And then the rest of this allows the lichen to kind of stick to its thing. Now, lichen are classified according to what they're capable of sticking to, right? So crustose lichen grow flat on the surface, crusty, right? They look crusty, right? And they're very common on rocks. Folios have these leaf-like projections, right? So this is something we'll often see around here, right? I have some growing on my fence, just doing its thing. Sometimes I'm wondering, should I scrape it off? Because I don't like it, but it is photosynthesizing. It is producing things, right? So usually I just leave it, right? And fruticose lichen are these branched leaf, finger-like, projections, right? This long, and they look kind of like moss, right? And so, but they're not really, right? There's this, again, this combination. Moss is a plant. This would be this combination of a fungus and an algae. As with everything, we're going to talk about some of the diseases that they cause, but sorry, folks, lichen, they're pretty nice, right? They don't really do too much in terms of causing disease, right? In fact, many produce antimicrobial compounds. Some have been are in the works for things like anti-cancer, anti-HIV treatment as well. Traditionally, because of these really interesting colors that they produce, and in fact, some of these you can stick in water, and then the water turns red, even though it's green. It's kind of weird. Produce these pigments that are used to dye wool. And then they're often been used as indicators of air pollution because they're so sensitive to things, right? When lichen are able to flourish, we say that we think that the air is good, right? But as a lichen number starts to decrease, it's kind of an indicator that the air pollution is starting to increase, and it's really affecting the lichen population. Maybe the most, let's call it bad lichen is Litharia vulpina, right? And it's somewhat toxic to mammals, right? Whatever that means, right? So it's not really deadly. Humans, maybe you'll get a little bit sick, but it's really not a big deal for humans. But traditionally, they were used to trap things like wolves and foxes, right? So way back in the day when we used to live in villages, and then wolves and foxes would come and eat your chickens and your goats and things like that, right? They'd lay out these little traps. They'd put out, dead goats or, meat, and then they'd lace it with Litharia, right? And then the wolves and the foxes would eat it and then hopefully keep them away. Okay, so that's enough for eukaryotic microbes. If you want to take a break, now will be a good time to take a break. as we shift gears to viruses, right? So viruses are going to come up next. We'll talk about viruses, and then that'll kind of finish off our whole other microbes thing, right? If you don't want to take a break, or if you're ready now, let's talk about viruses. So I say this for pretty much all microbes, right? But viruses are super cool, and like we're going to get into these even weird kind of virus things that are like insane, like blow your mind kind of stuff, right? But really, viruses are these things, they're not considered living, right? And they're like this, what they call this aggregation of non-living chemicals, which are inert. They don't really seem to do much. They're metabolically inactive. But you put them in the right cell, you put them in the right situation, all of a sudden, this virus seemingly comes to life, right? Even though it's not living. And it forces this cell to do these things and make many, many copies of these viruses, right? So it's really cool, really neat how, the way viruses work. And again, some of these other virus-like things, it's just really interesting how, we always say biology just kind of finds a way, right? So, let's start off with this general list of characteristics of viruses. And I think this goes back to our first module. Remember, viruses are acellular, right? They are not cells. And we talked about the list of things that are cells, right? They have to, they contain ribosomes, they contain membranes, they contain this and that, right? Viruses don't contain any of these things, right? So they're not cells. So oftentimes we refer to them as particles, right? So particles, later we'll learn this term called virions. And as such, viruses are not on the tree of life, not living, right? They're especially small. You cannot really see them using a microscope. You need something called an electron microscope to see it, right? So that's the only way we've ever really seen viruses. And they can't be filtered from solution, right? So we don't have filters with holes small enough to capture viruses, right? So that's kind of our definition of what is small. Bacteria, we can filter them, right? We have filters that can catch viruses, not for viruses. Bacteria, but not for viruses. Viruses contains a single type of nucleic acid. Here we have DNA or RNA, right? Remember, what do we have as cells? We have both, right? We have DNA and RNA, right? Of course, we use them for different things. Here we have things that have a DNA genome or an RNA genome, right? Which we always think is weird because we said genetic information, that's DNA. Well, viruses don't necessarily have to use DNA. In fact, they do really weird things, as we'll see later. And then they are surrounded by a protein coat called a capsid that surrounds this nucleic acid, right? And then some of those are enclosed by an envelope, which is sort of like a plasma membrane. And then that's it. That's what a virus is, right? So pretty simple for the most part. All viruses are obligate intracellular parasites, which means all viruses are infectious. No such thing as a non-infectious virus. There's no metabolic activity. So when you look at a virus, when you measure it, it really doesn't do anything, right? But when you put it inside a living cell, it'll multiply, right? It'll multiply, it'll force the cell to do what it needs to do. And then, in order to distinguish the virus from its many stages of the life cycle, right? Because you can have viral RNA or DNA floating around, you can have viral parts floating around, right? None of which are infectious, right? Only when it's fully synthesized, when it's fully put together, is it fully infectious, right? And that's what we call a virion, right? So sometimes we'll see this term a lot, not virus, but virion. And virion means that it's fully infectious, fully capable of doing its own thing. Viruses very rarely carry enzymes. Some have no enzymes. Some have one or two, as they're completely dependent on host cells, which actually makes treating viruses difficult because since they're going to use your ribosomes, since they're going to use your enzymes, if we want to stop them from replicating, we are in many ways going to stop ourselves from doing normal cell functions, right? So drugs that target the replication are also going to target us, right? So there has to be different ways of using therapeutics, right? If viruses don't have their own unique enzymes. Something that's not always understood is that all cells can be infected, even bacteria. We think of bacteria, these little guys that are going to go and do things, cause infection, right? But they can also be infected by viruses, right? So all cells have some sort of virus that can infect it. Now, this infection is limited to what's called a suitable host, right? So a virus can infect all cells, right? So just because all cells can be infected doesn't mean each virus can infect all cells, right? So we have this term called a host range, which is the range of host cells a virus can infect. And almost always, typically, it's one cell type of one host species, right? So viruses are very particular in what they're capable of infecting. And typically, this is determined by things like the virus's ability to attach and the availability of host cell factors for multiplying the virus itself, right? So, it's not that the virus doesn't want to infect, right? But if it can't stick, it's not going to be able to infect. Or if it can stick, but the host cell that it's stuck to doesn't have the right machinery to be able to replicate the virus, it's not going to work for the virus as well, right? So this viral host range, this narrow host range, gives potential in disease therapy. Imagine something like bacteriophages, which infect bacteria. And again, they're not going to infect all bacteria. They're going to infect very specific bacteria, sometimes specific strains of bacteria. So we can actually treat bacterial infections using bacteriophages, right? And then it doesn't affect the other bacteria that are around us. And of course, it won't infect us. Oncolytic viruses, the word onc, or onco refers to cancer, right? So, and remember, lytic for lysis, right? These are viruses that can lyse or break tumor cells, right? So oncolytic viruses can be used to, again, specifically infect tumor cells without affecting our normal cells. When we talk about viruses being small, there are, let's just go really, really small, right? Here are the kind of limits of the light microscope. This flu virus, and this would be like an ginormous flu virus. It's, basically at the super tiny limits. But really what we're talking about is, we'd have to have some super special light microscopes to see these things, right? So typically, this is kind of the area in which, most light microscopes work, right? Smaller than that are electron microscopes, right? And then oftentimes, in order to be able to see anything in this range, requires special coloring, special tagging, and things like that, right? So typically, we saw they're really small, much, much smaller than bacteria, right? Much, much smaller than parts of the cell. Like we said, all viruses consist of a nucleic acid, right? Remember, RNA or DNA surrounded by a capsid. And at its base, this is what a virus looks like. Nucleic acid surrounded by some sort of protein coat, right? So here we have an example of a nucleic acid. In this case, this would be DNA, and then this would be its protein coat, right? Each individual protein in the coat is called a capsomere. Two general types of viruses, we call them non-enveloped or naked viruses. Basically, it's just a capsid with its nucleic acid, and then enveloped viruses, which is surrounded by a lipid membrane, right? So here we have its nucleic acid. Here's the capsid that surrounds it, and then we have lipid bilayer envelope, which is essentially a plasma membrane that it got from its host that it infected, right? And then viruses often have these things called spikes, which are typically glycoproteins, right? Proteins with carbohydrate moieties stuck on the structure, and they're typically used either to bind to the host or to be released from the host, right? So they can do both, and they'll need both the spike to stick to and sometimes to leave the host as well. Now, when we put viruses together, when viruses are made, they can put their viral structure or the capsid together slightly different. So helical viruses are cylindrical or rod-shaped, right? So they have this long rod. Typically, they look like sticks right here. So it's tobacco mosaic virus. And just these individual sticks that you see are the tobacco mosaic virus. And they can be rigid like this, right? So they are these long, very stiff rods, or they can be flexible. So Ebola virus is an example of a helical virus that's flexible, right? So it looks kind of like a snake. Polyhedral viruses on the other end, poly meaning many, hedral for sides, many-sided, right? So we have something like this. Again, here's our nucleic acid, the capsid. In this case, we have many, many shapes kind of put together. And the most common shape is this thing called an icosahedron, which has 20 sides and 12 corners, but other shapes exist as well. And then these viruses can either be enveloped or non-enveloped, right? So you can actually have a helical envelope virus or helical non-envelope virus, polyhedral enveloped or envelope polyhedral or non-envelope polyhedral. This covers most viruses. There are some things called complex viruses where there are other structures attached to a capsid, right? So here's our capsid, once again our genome, and then sometimes there will be other things attached to that. And typically these other things are used for stuff like attachment, stuff like DNA delivery, right? So these tail fibers, this is a bacteriophage that infects bacteria. So these tail fibers are used to attach to the bacterium. The sheath is almost like a syringe that punctures the bacterium, and then that sends or allows for a tunnel for the bacteriophages to enter. Okay, so these are complex viruses, other structures attached to that. We do classify and do have a taxonomy of viruses. Generally, we don't use genus and species names because of the fact that they don't really like to be put on the tree of life, right? So often we refer to bacteria via families and genera, right? And these are based on things like their genetics, strategy for replication, as we'll talk about, their shape, their morphology, their biochemistry, and that, the types of molecules that they're composed of. General rule, and this kind of is a rule for all nomenclature, is that family names end in viridae, and then genus names end in virus, right? And then sometimes, usually they are italicized given that they are Latin and they are classical names. Species names can be used, but typically when they are used, they are often a common name, right? So it's some sort of descriptive common name, and then any subspecies can be designated by a number. So for example, we have, and don't memorize this list, please, right? I just want to go and show you some of these things. Often you'll see something like herpes viridae, right? The viridae indicates that we're talking about a family of viruses. Retroviridae, a family of viruses, right? And then it's a common name, right? Lentivirus, alphavirus, rhinovirus, sorry, the genus name, right? So these will be genus names, and you can tell because of the fact that it's italicized, right? Now, when we talk about something like, let's say, Ebola virus or influenza virus, right? So here we have influenza virus, right? That's this genus name because it's italicized. The common name for influenza virus is influenza virus, right? So that's its species. And then we can designate it by influenza A, B, or C, or some sort of strain number. Same with HIV, we HIV1, HIV2, et cetera, et cetera, et cetera. So we'll talk a little bit about the life cycle of viruses. We'll start off with prokaryotic hosts, and here's our proud parent moment. This really cool picture was taken by one of our students. We're studying some bacteriophages that are capable of infecting Pseudomonas aeruginosa. And so we took this to the electron microscope lab here at Manoa, and then they happened to catch this virus just as this cell, just as these viruses are being released. Right, so bacteriophages are viruses that infect bacteria. And then we have two different life cycles. One called a virulent phage that leads to violent death through lysis, like we're seeing right over here. And then something called a temperate phage, where the phage becomes part of the host DNA. And then whenever the host replicates, the virus gets replicated as well. However, eventually the cell is induced to make new viruses, these progeny virus. So then this temperate phage will eventually become a virulent phage, as we'll talk about. The viral life cycle can be quite complex. We're going to try to keep this as simple as possible. Try to use this general broad brush, right? Starting with the lytic cycle for lytic phages. All virus life cycles start off with attachment, right? So we have attachment where the phage attaches to the surface of the host. And for bacteriophages, they're going to use these tail fibers. And then we have penetration, whereby the phage DNA gets injected into our bacterium, right? So first things first, attachment. Then we have insertion of viral DNA into the host cell. And then we have biosynthesis. And in biosynthesis, what's going to happen is that we're going to take all these parts. Here we have our phage parts. Here we have our phage nucleic acid, right? All that stuff gets made, right? So the cell basically has stopped doing what it does, and it's completely turned over into this virus factory. During maturation, everything is put together. Now we're assembling new phage particles, right? Now we're creating these new virions, and then lysis, cell lysis. All these phages on the inside then are released, and then these will go on then to infect. These will then go on to infect our new host cells, right? And when it comes to things like phage therapy, where we can use phages to treat bacterial infection, lytic phages are the most useful because of the fact that they immediately destroy our cells, right? Now, temperate phage undergo what's called a lysogenic cycle, right? It starts the same. We have attachment, we have penetration, right? However, after penetration, the phage genome or the phage DNA becomes incorporated into the host genome, right? So now it becomes part of the host cell, right? This blue part is the phage DNA, and then whenever the cell divides, the phage is going to get copied, right? And this is what's called a prophage, right? So the prophage is that right there, right? And so the prophage DNA is going to be passed on to our daughter cells every single time the cell divides, right? Sometimes the phage brings with it some interesting properties, right? Some other genes, right? And in that case, that's called lysogenic, or sometimes called phage conversion, right? But typically it's called lysogenic conversion. And that's a change in the cell due to the phage, right? And there are some pathogens actually that have its ability to cause disease because of the fact that it has a prophage. And this prophage brings with it things like toxins and things like other factors that allow this bacterium then to infect, right? And then this host cell would then be called the lysogeny, right? At some point, the prophage pops out, right? And then now we go and continue our life cycle, right? So now here we have biosynthesis, where we're going to make the parts. We have maturation, where everything is put together, and then lysis, right? So then the whole life cycle then completes. These are then going to go on, infect our next cell, and then the cycle will continue. Okay, so that's your basic life cycle. We're going to just keep it that simple. Typically, phages will have DNA genomes, right? It's rare for them to have any other type of genome. So typically, they're DNA genomes undergoing some very simple life cycle: attachment, penetration, biosynthesis, maturation, release. Every now and then we have lysogeny, right? Or the formation of a prophage if you're a temperate phage. Animal hosts are similar, right? So the animal viruses, sorry, did I say animal hosts? Animal viruses have a very similar life cycle to a lytic phage, right? So our lytic animal viruses are similar to a lytic phage. They differ by some very specific mechanisms, right? Most specifically, this thing called uncoating, right? So actually, if we look at this, it looks very similar. We have attachment, right? So it's got to attach. We have penetration, right? In this case, penetration is the virion makes its way into the host cell. And then we'll have uncoating. And uncoating is this release of the nucleic acid from the capsid, right? So the capsid gets shed. Now the nucleic acid comes in, right? So that's different from phages, where phages will inject their DNA in. This doesn't quite work like that, right? The virus enters, and then it needs to be uncoated, right? After uncoating, very simple, similar stuff. We got some biosynthesis. In this case, assembly is maturation, and then we have release. Because of the fact that animals are a lot more complex, right? Typically, viruses will only be able to infect a certain animal, right? But also a certain tissue. And this is what we call tissue tropism. It's a tissue-specific infection of a host cell, right? So, for example, influenza virus really only infects the respiratory tract. If it comes in another way, if it doesn't have access to the cells of the respiratory tract, the influenza virus will not be able to infect you, right? Same with poliovirus. It's a central nervous system infecting virus, right? So, breathed in, it may not get access to where it needs to get to, right? So it's very important that viruses not only make its way to the specific host, also the specific tissue. Now, viruses really, animal viruses tend to be a lot more complex than bacteriophages. And a lot of this complexity lies in the biosynthesis phage. And the reason for that is because animal viruses have this incredibly diverse array of genomes, right? And by that I mean, we of course have what we think of as a normal genome, our double-stranded DNA genome, right? That's what we have. That's what bacteria have. Double-stranded genomes. We speak that language, right? But animal viruses can also have some really strange things like single-stranded DNA, which is kind of weird to us, right? But they can also have RNA genomes, right? Single-stranded RNA, double-stranded RNA, right? And then to make things even more complicated, we can have what's called a plus strand, single-stranded RNA, a minus strand, single-stranded RNA, right? And that's really talking about which side of the genome that it is. Is it the side that it's going to use, or is it the side that looks like or complementary to the side that it's going to use? RNA viruses then bring an extra complexity, right? Because we're not used to doing things like copying RNA regularly, right? So typically, during the biosynthesis stage, RNA viruses often have this RDRP, or something called an RNA-dependent RNA polymerase, right? And this is a viral enzyme, a viral encoded enzyme, which means it comes with it in its own genome, that then can take this single-stranded RNA, turn that eventually into double-stranded DNA, right? Copy itself, and then that eventually gets turned into double-stranded or single-stranded positive sense RNA, which then can get converted to a host ribosome, used by our host ribosomes to make viral proteins, right? So this RDRP is very important, so much so that it's often the target for therapeutics, right? Many therapeutics will target the viral specific enzymes, right? Because if we can knock out and target this RDRP, then the virus will not be able to copy itself. Retroviruses are a special class of RNA genomes. RNA viruses, right? Retroviruses are these positive strand, single-stranded RNA genomes, right? And then what they do, aside from having an RDRP, is this thing called a reverse transcriptase, right? So whereas the RDRP is used to take the RNA genome and get copied into more RNA, right? So then through the biosynthesis, reverse transcriptase does the opposite. It takes this single-stranded RNA genome and turns it into double-stranded DNA, right? So here we have HIV, which is our classic retrovirus, right? And then let's go. What is this? What part of the life cycle is this? Number one, that's attachment, right? Good. And then we have entry, right? Or penetration. So we have the entry of the virus, in this case, the capsid. And then now we're stripping the capsid away. This is called uncoating, right? Good. So we have uncoating, and then now our single-stranded RNA gets turned into double-stranded DNA. And then just like with our lysogenic viruses, this double-stranded RNA now becomes part of the genome, right? And this is what we call a provirus, right? Prophage for our bacteriophage. In this case, now it's a provirus, right? So now whenever the cell copies itself, the virus gets copied. More importantly, it hides from the immune system. It hides from everyone else because it's just DNA stuck inside the cell somewhere. And again, after quote unquote some period of time, right, the provirus pops out, and then now we start to undergo biosynthesis. We start to make and maturation, start to make mature virions, and then we now leave the cell, and then they can go on and infect themselves a new cell. Okay. So the type of viral life cycle then lends itself to the type of viral infection, right? And most viruses cause what's called an acute infection, right? It's this short viral infection, right? The way I like to call it is it's acute infection. Cute, short and sweet, right? Basically because it just undergoes its lytic life cycle, right? It's going to infect, stuff is going to happen, it's going to get copied, biosynthesis, maturation, right? Release. And then either it's going to win and we die, or hopefully our immune system will win and then the virus is eliminated, right? And something like the cold and the flu are what we call an acute infection, right? Short. Persistent infections are long-term infections due to an inability to clear viruses, right? And that's because we're unable to clear it, either because we lack the mechanism to do so, right? Or because the virus hides, right? And so there's two general types, one called a latent infection. And in a latent infection, the virus remains hidden for a long period of time, right? So if we look at this on a graph, right, you know, we could have something like an acute infection, right? This is over time. All right, we have that's a T. That's a T. We can have the virus replicates we start to beat the virus is gone. That would be an acute infection, right? In a latent infection, it would be no virus or no virions for a long time. And then at some point, we then have our, it pops out, it starts to replicate, right? And then again, things happen, right? So, but typically you are symptomless for long periods of time. So something like shingles is a latent infection in that, we'd have our initial chickenpox infection, and then the virus just kind of hides somewhere, right? We don't see it for a long time, and at some point when it decides to, it'll pop out, and then you get yourself your little shingles, right? As opposed to a chronic infection, which is this long persistent infection, right? Where you have this gradual buildup, oops, that's a little high, of virions, and then once again, we either, it pops to life and then we clear it, or it pops to life and we die, right? So HIV is an example of a chronic infection after a period of latency, right? So actually, HIV is kind of a combination where the infection will always have this acute phase, right? It'll be quiet for a long time, and then this gradual chronic infection, right? And usually at that point, it's kind of not good for you, right? And chronic infections tend to be real bad because it slowly weakens the immune system, and makes you more and more susceptible to things. So I do want to talk about plant viruses just a little bit. I think my botanist friend would not be happy if I didn't talk about plant viruses. For the most part, very similar, right? They're going to go through the exact same life cycle. Genetically, they're not quite as diverse, so most of them have a positive strand, single-stranded RNA genome. And a little different in that viruses tend to have this really broad host range. Some plant viruses can infect thousands of plants. It's crazy, right? And generally, plant viruses tend to be what's called biotrophic, in that they infect without killing the host, right? So it does cause damage. It does do a lot of things, right? Generally, it's not fatal, right? But over time, the plant gets all weak and weird looking and all that kind of stuff. And generally, it causes a lot of problems, especially if it's a commercial crop, right? So I think one of the more famous ones around here was the papaya ring spot virus, which really decimated the papaya industry on Hawaii Island, and then which then led to the introduction of the genetically modified papaya, right? So there's a really cool story about that, the rainbow papaya. You guys can look it up on YouTube, I believe, right? Papaya ring spot virus. So really cool stuff. And so that's the main reason I wanted to talk about plant viruses, right? Even though they don't really cause too much trouble in the sense that, obviously they're going to infect humans, right? But they have this huge economic impact, right? Because anything that targets commercial crops can really decimate the industry. One super quick look at the viral growth curve, right? So here we talk about what's called a one-step growth curve, right? Which measures the amount of virions present after one round of viral replication, right? And this allows us to do a few things, right? So first of all, here we have initial infection, right? So the number of virions, and then this drop in virion number, and this would be free virions floating around, right? So why do you think we go from this infection to all of a sudden drop in virion number? Where do they go? Yeah, that's right. They have to go inside the cells, right? So we're always going to be infected by a certain number of virions, and then they're going to drop, right, as they enter the cells. And then it's during this time that we undergo this biosynthesis, maturation. If we have this latency or the lysogeny, that's all going to be there. But at some point, it's now going to go into the release, and then we see this release, right? So what ends up happening is that as we have infected the cells, all the cells are going to release the virions, and then this is what we call the burst. As the cells burst, right? Pretty clever name. And then the virions are released. And then at the end of that, we get this plateau, right? As the virions get released. What's going on with my pointer here? As the virions get released, and then, all right, there we go. As virions get released, and then we have this this plateau, right? And this is what's called the burst size, right? The number of virions from here to there divided by the number of cells indicates how many virions are released per bacterium. Okay, and that's pretty much all we want to talk about viruses. Now we're going to get into like the weird stuff, right? Viruses are already pretty weird, right? Now we're going to get into the weird stuff. Stuff like these things called viroids, virusoids, and prions. And like this is like weird, right? Like we talk about how biology always finds a way, right? Life finds a way. Well, I mean, this is like really weird, right? Basically, they're non-living disease-causing agents, like viruses, right? But they're different, right? However, just like viruses, they do require a host to be replicated, right? But the structures are so different, and the fact that they're even able to survive, and where do they come from? It's like really crazy stuff. Viroids are short strands of circular RNA. Right, let me repeat that. They're short strands of circular RNA. And what is up with that, right? These things are capable of self-replicating, right? However, they do require host machinery, right? And there are many plant viruses, plant pathogens, these viroids that are capable of infecting, right? So like this potato one, for example, right? It's just a short strand of circular RNA that infects this potato, causing this disease, right? There's no protein coat, no nothing else. They can be self-replicated, but they require host machinery to do that, right? Weird stuff, I know. Even weirder are these things called virusoids, right? Like who names these things? Virusoids are non-self-replicating single-stranded RNA. So now we have these things that are capable of infecting, that are single-stranded RNA, and they don't even replicate themselves. It requires this thing called a helper virus. Like what? It needs another virus for it to be able to replicate, right? So there's this hepatitis delta virusoid that requires hepatitis B virus, right? So those that are infected with hepatitis B sometimes get infected with hepatitis delta virusoid, and that increases the pathology of hepatitis B virus. But if you somehow acquire hepatitis delta virusoid, you're not going to get infection because it requires this hepatitis B virus in order for it to be replicated, right? So it's crazy, right? It's this infectious RNA, right? That's non-self-replicating single-stranded RNA, and that's all it is. And yet somehow it can cause this debilitating disease. Even weirder than that are these things called prions, and prions are these infectious proteins, like proteins that are infectious, right? That's crazy. And what they are these misfolded form of a normal protein, right? So we have totally normal protein, and then when we have this prion, right, it now interacts with our normal protein, and then undergoes this conversion, and it causes them to become this misfolded or this mutant form of this protein. And not only does it do this, it's transmissible, right? So the most famous case of this is this transmissible spongiform encephalopathy, right? AKA mad cow disease, right? In humans, it's called Creutzfeldt-Jakob disease, right? Where it starts to take brain tissue, right? And then as the proteins start to interact and replicate, it then causes these lesions, these sponge-like lesions in the brain tissue, and this degenerative neurological disorder, right? It's crazy stuff, right? So, I always thought viruses were really cool in the sense that they're really scraping the edge of what is living, what is non-living. We always think life is all about figuring out a way to replicate. Well, these virusoids, prions, right? Viroids, are they also living? Because, really, at the end of the day, what are they trying to do? They're trying to replicate themselves. And somehow they figured out how to do that, right? Anyways, mind is blown. I need to take a break. So that's it for Module 3. If you have any questions, right? I know a lot of cool things out there. We just kind of scraped the surface of a lot of these things, right? So there's a lot more reading that you can do, right? I encourage you to go and learn more about these crazy things out there and all these different types of microbes that are not bacteria, right? Lots of interesting things out there, right? So much to learn. Until then, we will see you in Module 4, where we talk all about cell metabolism.